Alright, in these review videos, what we're going to take a look at is the whole idea behind the circulation, the recycling of carbon throughout the carbon cycles. And when doing so, we're going to be talking about two main processes, those being, or that being, cellular respiration and photosynthesis. So if I take a look at the basic byproducts of what I need in cellular respiration, and so what we need to take in, that it being O2 and glucose, C6H12O6. What I am going to make out of that is a waste product of carbon dioxide, CO2. Let me shrink this up a little bit. And water, H2O. So that is going to be our waste product. So CO2 and water coming from this being our products. This is then, or excuse me, our reactants and then our products. Now this process we have as cell respiration. Why do we eat? Why do we breathe? Is in order to get this waste product to make energy in the form of ATP. So if we break those down into the basic processes, am I going to start splitting up the carbon dioxide, and then where is water going to come into play in this process? So if I go through and I can start taking a look at the process, the first step that we're going to look at is glycolysis. Glycolysis, really simply stated, means the breakdown of sugar. So I'm going to take glycolysis and I'm going to take our equation of C6H12O6. There's our sugar. What I want to do with this sugar is I want to break it up, and in order to split it apart, I need to expend some sort of energy. So here's our reaction. What I needed to do is take in some energy. In fact, I need to take in 2 energy units, and our energy unit we're looking at is 2 ATP. So what I actually get out of it are 2 ADPs. So cleaving off of a phosphate, that cleaving off of that phosphate is going to give us the energy. What I get in our intermediate step is a structure, a 3-carbon structure, that is going to be utilized to make pyruvate. And that 3-carbon structure looks something like this, with a phosphate group on it. Now, being that this is 3-carbon structure and we have C6, we actually make 2 of them. So we have another one. Now, what these guys are called are PGALs, phosphoglyceraldehyde, or G3P. Those structures there are the intermediate step to get what our end product we want as a result, pyruvate, and those are just a 3-carbon structure. Now, so each one of those is going to get us a 3-carbon structure called pyruvate. Each of those are a pyruvate structure. Now, what do I actually get out of here? In order to do this, what I get out per G3P or per PGAL is going to be 2 ATPs. And more importantly, what I'm going to get is an electron. And my electron carrier is going to be nicotinamine, and this H is the reduced form. That's our electron that can be working with. So we're going to call this from here on out a bus, or our little carrier, and this is our NAD. Now, so this is per pyruvate or per PGAL, we're going to get this. So if I'm going to narrow it down at the very end, what do I end with in this process? In glycolysis, I am going to go from PGAL to pyruvate, and what I am going to net is a total of, in glycolysis, 2 ATP, 2 NADHs. So that's what I'm referring to at the end of glycolysis. Now, with that pyruvate, the end products here, which is our 3-carbon structures, 1, 2, 3, we are going to move on and take this 3-carbon structure into the mitochondria. That's where if this oxygen is present. If that oxygen is present, we're going to go to the mitochondria. If it is not, we are actually going to take it and leave it in the cytosol with the anaerobic respiration, and we can undergo one of two steps. Those one of two steps are the fermentation. Fermentation process can be one ethanol, which is like ethyl alcohol, or number two would be lactic acid. So those are the two forms that this guy could undergo under the fermentation process: ethyl alcohol or alcohol fermentation, or lactic acid. Now, in order to do that, what we're taking this pyruvate, and we're going to get ATP from it. Okay, 1 ATP per, and then from there, 1 ATP per that we're going to get is 1 ATP, and we're going to get a return or oxidized form of our bus. So we had NADH. After glycolysis, this NADH is going to then return back to this NAD to be our carrier again. So in this process of fermentation, we are getting per ones of these 1 ATP and then 1 NADH to become oxidized again to become NAD. So if it stays in the cytosol, we are getting, after for the fermentation process, 2 more ATP and then 2 NADs that have now been returned. Okay? If oxygen is present, we then go to a different cycle, different step. We are going to go into the mitochondria. So this is our mitochondria. Inside there, we've got, it's called the cristae, these inner folds that I'm creating. Okay? So this is your mitochondria. This structure here is going to enter in. And as it starts to enter into the mitochondria, it actually has a carbon being cleaved off. And that carbon getting cleaved off will leave in the form of carbon dioxide. What's left is a 2-carbon structure, this 2-carbon structure that we are now going to call acetyl coenzyme A, or acetyl-CoA. Now, in the process of doing that, I am actually going to get another electron carrier, that being NADH again. So we can get one more of these. But acetyl-CoA is going to be in this space. In that space, we're going to call the mitochondrial matrix. So that's the matrix. And in the matrix, we're going to go and do what's called the Krebs cycle, where I take acetyl-CoA and I am going to join up with a 4-carbon structure of oxaloacetate. This collectively, when done, is going to make a 6-carbon structure. So a 4-carbon plus a 2-carbon. So there's our 6 carbons of citrate. Okay. Now, this cycle, again, is called Krebs. It is also called the citric acid cycle. So the tricyclic acid cycle. Now, what's going to happen is that we have this acetyl-CoA. Its job is to go basically in a circle to make this oxaloacetate again. In the process of doing this, we're not going to worry about these intermediate steps. We're going to talk about what are we getting out of it. Now, per acetyl-CoA, we only get 1 ATP out of it. Not very efficient. So per glucose, we get 2 ATP. Per acetyl-CoA, you only get 1. But what we do get out per acetyl-CoA are 3 NADHs, so 3 more electrons, and 1 FADH2. These are our buses that are going to carry the electrons. Now this one you can think of as a double-decker bus because it can carry 2 electrons. So its job is to take those electrons, and it's going to move into what we're going to, our last step, called the electron transport chain. Now, if this, what we have in here, is per pyruvate, then all you have to do is times it by 2 to get it into per glucose. Okay, so we end with 2 ATP, 6 NADHs, and 2 FADH2s. Ultimately, what we're then going to end with is these electrons are going to move their way to the electron transport chain that is happening, if I go back real quick, inside of this guy here. So if I'm going back, this structure is the cristae. So these folds of the inner mitochondrial membrane are the cristae. Move over to the next slide, you can take a look. This is the fold of the membrane inside the cristae. What I'm creating now is just a quick phospholipid bilayer within this. Okay? Now, up here, there's the outer membrane of the cristae. This is the inner membrane, so in here is the matrix. This is the space between. So if I look back at this, this is the space. Not outside, not inside, not in the matrix, but the space between. What's going to happen is that all those electron carriers that happened in the matrix, those 6 NADHs and 2 FADH2s, their job is to work through and create what's called this electron transport chain. Now, in order to do that, we are going to, I'm just going to remove this real quick. And we had 2 FADH2s. Its job is to come in and work through the start of this protein. As it works through this protein, what it's going to do is it's going to take this hydrogen. You can imagine this hydrogen structure. We've got a hydrogen, here's the nucleus, and around it is one lonely old electron. This lonely old electron is going to be there. And so this, we're going to color it just shaded in blue, is the proton. So that's the positive portion of it. Okay? So you think of it like as just a plus sign. There's our positive portion. Now this protein up here is going to take that electron, grab it, and shuttle it on through this membrane. What's left is this big structure there. We'll just draw it in a different color. This positive gets shuttled up through. So if I'm going to illustrate that real quick, you see your phospholipid bilayer on either side. And here is a hydrogen ion, or hydrogen. The electron that's around it will eventually, let's actually give this a different color so you can see it. So that electron will be cleaved off, and the electron will start working through this chain. Now this hydrogen will be left is the hydrogen proton will be shuttled up through. Now when I have a whole bunch of these protons starting to be shuttled up through, I start creating what's a gradient. So I've got a lot of these H's up here. These hydrogen protons that are going to be up on the other side, upper surface of the cell membrane. Now, if I continue working on my cell membrane, I'm going to have another protein set. But as I create a high concentration to low concentration of those, I am going to work my way through another protein, and the base of this protein has this big channel on it. You can think of it as like a big wheel. As these hydrogens work on through, it churns this wheel. It's continuing to churn this wheel, and what I have are inorganic phosphates with an ADP that are going to be coupled together. So what this is now this electron transport chain of these electrons going through, the protons being pumped up and then coming back down. Now, when it comes back down, I am getting this inorganic phosphate. So this is what we call oxidative phosphorylation. And then this last electron that's being pumped through, it's going to be accepted by this last electron acceptor, that being water. So when I have, or I should say oxygen, so I have an electron that's going to have my hydrogen that will come back on through. When I join it up with oxygen, I eventually can join up with a couple of them, and then I utilize those two things to make water. That's it for cellular respiration. We'll move our way into the photosynthesis next.